So good evening. Welcome to our virtual MADIA meetup for February. Hope you're all safe and well. I'm Bruce Lathrop, a member of the MADIA board. I'll be introducing our presenter, but I first wanted to thank Brenda Trainer, who's helping with this meetup, and Jeanette may jump in as well. Brenda will be moderating the chat and live questions. Our presenter will be answering questions after his presentation. For those unfamiliar with Zoom, you can click on the chat at any time on the bottom of the screen, as Brenda said, and put in your questions, and she'll talk to the presenter about them. At that time, the presenter will also be taking questions. You may be muted now, so unmute yourself and ask your question. Before we start the presentation, I wanted to tell you a little bit about MADIA. We're a 501c3 educational organization centered at the heart of San Gabriel Valley in California, and our mission is to ensure the region's technical, scientific, and industrial workforce is connected and has the tools they need to be successful by providing educational programs in entrepreneurship, business finance and operations, and technology development and communications in settings which encourage networking and collaboration among our members. Our next meeting will be part of the MADIA Manufacturing Week, which will run from March 1st to March 11th. Exact dates will be announced. One of our presenters will be Darcy, who will be speaking with Thomas Me about their company, Irwindale-based fog cooling company Me Industries. If you have any feedback on this program or suggestions, please send your comments and ideas to email at madiatech.org. Stay online after the presentation and and grab your favorite beverage, and we'll continue networking. I'll now turn to introducing our presenter. Dr. Tyler Gorman is the Director of R&D for Invizyne and has been a central player in developing Invizyne's patent portfolio. He earned his BS and MS in chemistry from UC San Diego and his PhD in biochemistry at UC Irvine. At UC Irvine, Dr. Gorman worked on the structural biology of polyketide biosynthetic enzymes in the lab of Dr. Cheryl Say. From 2008 to 2019, Dr. Gorman worked in the Bowie lab at UCLA, where he pioneered cell-free methods of producing bio-based chemicals. While at UCLA, Dr. Gorman had led several small teams focused on applying cell-free biocatalysts to production of a diverse range of natural products, from biofuels and bioplastics to terpenes and polyketides and cannabinoids. Dr. Gorman was a Camille and Henry Dreyfus postdoctoral fellow in environmental chemistry from 2009 to 2011, and a recipient of the 2014 NDI Postdoctoral Award for Research Excellence. Along with Dr. Bowie and Paul Abiganorth, Dr. Gorman founded Invizyne Technology to commercialize their cell-free platform and open their doors in Monrovia in 2019, following initial funding by MDB Capital Group. In addition to raising the seed round, Dr. Korman is a principal investigator on multiple SBIR and government grants awarded to Invizyne Technology. So please welcome Tyler.
Thank you very much, Bruce. Let me share my screen. Okay, thank you everybody for showing up. Can you guys see the screen okay? I have a big black.
Yes, we can see it.
Yes. Okay. Great. So yeah, Bruce, thank you for the introduction. Happy to be here to present, maybe more of your college 101 biochemistry than any of you ever took. But hopefully it's not too much science. I'll basically tell you generally about some of the science and technology about what makes our technology work, and then kind of a story about how Invizyne was founded and kind of the path we've taken here in Monrovia as we've gotten up and running over this past year. So kind of the title of my talk, Biosynthesis Beyond Biology. We're really trying to usher in this next wave of bioproduction. A lot of people right now are you might be familiar with metabolic engineering using yeast. There's a number of companies using yeast and other type of microbes to make things besides ethanol. you know, drugs, different types of flavors and fragrances and so forth. We have a different approach. It's still bio-based, but we think it has potential to solve a lot of the problems that are presented with cells. So this general idea is that over the past 20 years, what a lot of researchers, companies have tried to do is to really turn a microbe into some type of cellular factory where you can just feed it sugar and, you know, allow that cell or those cells to make a variety of different compounds that can be used as flavors and fragrances, pharmaceutical compounds or intermediates, and biofuels. So that was the hope and dream. And it's great. It's a really simple idea. And there's, you know, a lot of promise there. And so the way that this typically is done, so the S is a substrate. So you want to turn that substrate into P as the product using this engineered microbe. So you can see the little DNA there. You know, let's say you want to take your substrate is glucose, a sugar, and you want to turn it into your product. That's limonene. So that's the compound that makes lemons smell like lemons. And the way that people would do this is, you know, they'd make an edit to the DNA in the chromosome in the bacteria. And a lot of times they'd put some kind of pathway on a DNA vector to then change that, enable that cell to now make their product. And so each of those genes encodes an enzyme that allows them to turn that substrate into some intermediate. And that whole pathway now links these enzymes together to be able to turn that substrate into this non-natural product that the cell doesn't normally make. And cells are really, really good at this because, you know, they have all the energy and things that, you know, power life to make these pathways run, right? So these are biological pathways that use this energy. And so, you know, it's a really great idea, but it really begs the question, why aren't there more commodity chemicals and biofuels being produced using engineered cells? There's actually really few industrial examples of this being used. So, you know, some obvious examples, ethanol is one good one. There's some amino acids that are made using microbes. Some organic acids like acetate, acetic acid are used this way. A lot of times they're used just by using a naturally occurring microbe that's maybe been engineered a little bit. You know, so what's going on here? Why aren't there more things being used using this engineering approach? And the answer really is that these cells are complex, right? So, you know, it's no different than any cells in your body, really. I mean, there's a lot of other things going on besides that pathway that you're putting in there. And so, you know, what this has the effect of doing is that you have all these unwanted or unknown outcomes because engineering life is really hard. It makes design, build, test cycles really long. And this whole process of engineering the cell, it's like engineering a black box. You don't really know what the outcome is going to be. It's difficult, slow, and it can be really costly. And so, you know, in practical terms, what happens is background metabolism, so the other things that are going on, lowers the titers and yields. So it sucks off that carbon to, you know, produce other chemicals that the cells need to live. So instead of having that glucose go to your product, it just goes to the other things. A lot of times the things that you want to make in a cell are toxic, right? Antibiotics, if you want to make an antibiotic using an engineered cell, well, that's the definition of that compound, right? It kills microbes. Sorry, I'm getting some background. And so that makes things really difficult as well because, you know, the cells don't grow as well as they can. You can try and engineer out these competing pathways, but a lot of times this has unwanted side effects. So you never, again, you're engineering a black box. You don't know what's going to happen. And so, you know, one day I remember it was about 2014, while I was at UCLA, we had done a variety of projects where we had tried to engineer microbes to make a variety of different things. Biodiesel was one of them. We were trying to make fatty acid methyl esters, and it worked okay enough to publish some papers, but it was never something that was going to be commercial. And I remember Dr. Bowie came up to me and he's like, well, what if we just got rid of the cells? So how do we simplify this whole process? And so that was our answer. What if we just got rid of the cell and, you know, reconstituted these pathways? You know, it was kind of a simple idea to a complex problem. And so, you know, we were just naive enough to say, hey, well, let's try it. And so what we do basically is we still use metabolism and, you know, all those enzyme pathways that make cells run. We use that as our blueprint. And then we can just design out, you know, literally searching databases to link these steps together You can do this on pen and paper. You can use more sophisticated methods like computational methods to design these pathways. But at the end of the day, you're really just linking a substrate to a product by a bunch of different steps. You can then throw out that blueprint, and now you just are left with the pathway that you want. So you don't have to worry about any of these other competing pathways. And it makes the engineering and the design really easy, because if you have another enzyme that's better for whatever reason, maybe it's more stable, maybe it's faster, you can just easily swap it out. And so, the next step in that is you now make these enzymes. So that's a relatively robust technology where you can take a microbe like E. coli, put in a DNA, and it will just make that one protein. So you make lots and lots of each of these individual proteins, and then you can isolate them. You can just heat, this batch of enzyme, and it kills everything else besides your protein of interest if you're using a stable enzyme. Then you literally just mix them together with the energy cofactors that you need and run it. And so it's really simple, kind of elegant way to do biocatalysis, where you're turning biological substrates into bioproducts using enzymes. And so it's not a new idea. So Buchner in the late 1800s, he was kind of the start of a lot of what's possible for metabolic engineering and all of this, bio-based things, actually. So he took yeast, and he showed that if you take a dead yeast and you just use the lysate, so those enzymes that you can recover from this dead microbe, and just feed it glucose, it'll make ethanol. And so that was the first cell-free example. And so we want to take that kind of idea where we feed our enzymatic system sugar, we break it down into these building blocks. You also get some energy in that process, and then you can link it to another enzyme pathway to make useful compounds. And so we want to do this all in one pot, and we want to have this run continuously for a long period of time. That would be the ideal. So you're just constantly feeding in sugar and having product come out the other end. One of the key components here, though, is these cofactors are really important. So they're the energy that basically drives this whole machine. And you can't just keep adding these cofactors. You have to recycle them because they have a definite cost. And a lot of times that cost is very high. Part of the problem also is that these biological systems are really fine-tuned so that there's bottlenecks if you don't make and use these energy sources appropriately. And so some of our key innovations that I did while I was at UCLA was we developed really slick methods to regulate this energy usage and to kind of break rules of stoichiometry. And so, one of the premises that we were working on was that, we wanted to have a system that you could just literally set up and walk away. So it was auto-regulatory, right? It had minimum intervention. And it would allow a system like this to run where, if you're breaking down sugar and you're making energy, so this is the NADP to ATP or NAD to NADH, that's your forms of energy. And the N is the number of molecules. So if you're making those and using those stoichiometrically, that's great, but it's really a perpetual motion machine. Eventually, if you have any kind of leakage from that system, the whole thing will shut down. And so, if ATP goes back to ADP and it's not being used, you're not going to make any product. And so our innovations, we gave them catchy nicknames: nuclear purge valve, molecular rheostat, kind of so we could publish a little bit. But they enabled us to now break these rules of stoichiometry, and they can just be dropped in. These are enzyme systems that enable us now to build up excess energy and still maintain all the flux to our useful compounds. So it was a really useful technique because it just dropped right in. There was no extra things that you needed to add. It was just an enzyme or two, and it allowed these things to function really, really well for long periods of time. It actually worked much better than we had ever dreamed that it probably could. And so, at any given time, there was only probably 2 to 3 people working on any of these projects. And within 5 or 6 years, we were able to apply this one basic premise to make a variety of different compounds. So we made not just biofuels shown on the right. That was our first target at levels much higher than you could do in microbes, because these fuels are like solvents. They kill the microbes as they make them. We made it at least 5 times higher than what you could do in cells. We're actually now another 5 times higher than than these levels even. So we've improved that further. We also demonstrated that you can make flavors and fragrances. So like that lemon smell, limonene that I showed you on that first slide. So we made that at really high titers along with the compound that makes in sandalwood oil, santalene. So we could do that really well. And then, at a certain point, we took a step back and we said, hey, we can make some of these compounds, maybe they're building blocks for other things. And that's true for this story of cannabinoids, where, the flavor and fragrance limonene shares a common intermediate with cannabinoid biosynthesis. And so we said, hey, could we use this technology which works really, really well and apply it to something that is really hot right now, and a therapeutically relevant class of compounds? And so that's what we did on the left as well. So Saken, who's on this call, and some other people, Paul Aquanorth, one of the other founders at UCLA, were really instrumental in doing a lot of this work and getting this, technology off the ground. And so it really, shows the power of not just good science, but a good idea. And so this is kind of highlighting some of the things that we needed to do because we were using enzymes and people know a lot about enzymes. They know the DNA that encodes for an enzyme, and you can then go back in and make specific changes to those enzymes. So we rely really heavily on this ability to figure out what enzymes look like and then change them around to change their product, their substrate specificity. So what kind of things that they'll accept. And so this is the case with this cannabinoid project that we had. So we wanted to make this compound CBGA, which is the precursor to cannabinoids. And so if you see here, so the enzyme that we were using makes this unwanted isomer here. And through some in silico design, so computational design and then combining those mutations, we now had a mutant that made almost exclusively this compound of interest. And so then we were able to design kind of a toned down pathway shown on the right hand here. One of our initial things that we did is we set up a 33 enzyme pathway, which for those of you that don't know, it's bordering on insanity. Like nobody would have really thought to have done that. And we did it and it worked, but it was a lot of work. So moving forward into the commercial space, we knew we had to simplify things. And so we've been relatively successful at that. So we can now use these pared down pathways where we can now make these building block precursors and then put in a variety of different inputs to make interesting molecules as well. And because this whole system is modular, we can easily swap out inputs to make other products. And so we think we have a really robust and flexible system to make a variety, in this case, of therapeutically relevant compounds. And so we're really excited about the future of this. We're currently in the process of scaling this as well as expanding it to make other interesting compounds. And so that's kind of the science behind it. And I'll go into my background a little bit and what brought us to Invizyne and how this works. So like Bruce said, I started off in the Scilab at UC Irvine studying these polyketide synthase enzymes. So you know what they are, even if you don't know what a polyketide is. So antibiotics, tetracycline, for instance, is a polyketide. So we were studying the enzymes that make those drugs in microbes. So a lot of these come from soil bacteria. And so I figured out, I used that to learn how proteins work and what they look like. It was more of a basic science problem. I then went into the Biolab in 2008 to work on a more applied project to engineer a lipase as an alternative for biodiesel production, where the idea was you just take methanol and olive oil, for instance, and this lipase will catalyze that reaction to turn that oil into a methyl ester, which is biodiesel. And so the lipase being a biological thing and not a chemical, tends to fall apart in the presence of a solvent like methanol. And so we engineered it to be more stable. Then came the kind of epiphany where we're like, hey, this metabolic engineering stuff is hard. Why don't we do these pathways? And so that was really great for me because it was a point where I could take my background in secondary metabolism, so making these natural products, and apply it to now a project to make really interesting chemicals. And so it was an idea. We ended up getting some money from the Department of Energy to actually apply this to fuel production. So these terpene molecules can be used as fuels potentially. So that was really what got the project off the ground. And with those initial papers, we thought we had something cool. And so we incorporated the company in 2015, which is, a blessing and a curse. So we were way too early. We didn't have kind of the hook that we needed to really attract, business and investment. And so it wasn't until towards the end when we had this hook of, being able to prenylate these molecules to make cannabinoids that really allowed us it was the final piece of the puzzle that allowed us to attract investment. Also, we had built up enough of a background of high profile papers and notoriety where now the government was starting to take notice. And so they were starting to write calls for grants that called us out specifically and said, okay, well, we should be applying for these. And so, within a month or two of us opening doors here, we ended up getting at least 2 grants, if not 3. And so, that really took off kind of the end of 2019. I started here with Paul, our co-founder, and we quickly hired John. And it was the 3 of us for a while until I think June or July when we started to hire again. So we've gone from 3 to 7 plus the CEO in the course of the pandemic, which I think is no small feat. In addition to raising money, we also, got 3 different grants. And so it's been really great. And, we started with this premise of, can we use, these isolated pathways to do things that people have done in the past using cells, but only better. And then take that and, apply it to a wide range. We really think we have a platform technology here, not just a single thing that we can use to make just one thing. We're constantly inspired for the need for constant development. I never thought that I would start a company. Actually, I was quite happy at UCLA in the eventually in the project scientist role, working in a place that was a great environment, doing really cool stuff. But we started Invizyne because we knew we had to move it off campus in order to do what we were doing. For those of you that a lot of you probably know, we were searching for commercial targets for a while. It took us a while to figure it out. And actually, one of the things besides the discovery and development of this prenyl transferase, which was the key cog for the technology, actually the biggest, probably one of the best things we did was by mistake. And I went to a conference up in the Bay Area, and being naive, there was somebody there that was from another company that was interested in licensing the technology. And I was like, oh, this is cool. We had already incorporated Invizyne. And I said, well, you should reach out to UCLA and say that you're interested in licensing it. And they actually did that. And so I was contacted by Tech Transfer and I talked to another professor, and he's like, what did you do? He's like, if you're gonna keep control over your IP, you need to get in the middle of this and, shut that down. And so in doing that, UCLA then said, well, we have somebody who's willing to pay. You guys need to pay. We'll stop, but you guys need to be serious about this. And so that basically happened about 2018, and we were real serious about in 2019, really trying to do all the things we needed to do to build this business and get it off the ground. And so we raised private money in early 2019, opened doors after a lot of searching for good places to be. And Oakcrest Institute of Science this incubator is fantastic. Like I said, we've also received a couple of SBIRs, one of them that's been bumped up to a phase 2, in addition to a really large grant from the Department of Energy, and hopefully more to come. So we have some other grants out there as well. I'd just like to highlight now, I'm almost right on time for 20 minutes is good. In terms of, the biotech industry in LA and the San Gabriel Valley, I think tech is quite strong around here. I think the biotech people are just not quite aware of it. You know, I think a lot of people will be surprised to hear that there's $1 billion company down the street in Monrovia. That's a, you know, Zencor, you know, that makes antibodies and drugs. It's just not publicized. You know, if you think about biotech in San Francisco, San Diego, Boston, there's a lot of hype around it. There's a lot of companies. And it makes a difference because those people that are looking for those jobs, you know that they have a skill set that can be easily translated to different projects. So if that startup goes under or they're not happy there, they can easily go to one or the other. And so just knowing what the options are around LA is sometimes really difficult. It also speaks to how difficult it is to recruit. We've been really lucky and really good about finding the right people. We've had the benefit of Corona allowing things to slow down a little bit so we could be more selective. You know, but you know, it helps that the people that you do get tend to be more invested. But you know, as you're growing, if you have to grow fast, it really leads to challenges about, you know, how do you actually grow fast if you have to, if the talent is maybe unwilling to relocate from San Diego or the Bay Area. You know, one of the things that I've learned is don't be afraid to talk to people inside and outside of your network. One of our chemists we hired just off of LinkedIn because I thought his job description sounded interesting. And I just reached out and said, hey, you know, let's connect. And then he connected and I said, you're looking for a job? And he said, yes. And, you know, here we are 3 months later. Funding through grants, you know, is great. They're offered to small businesses, you know, through all the different organizations, DOE, DOD. You know, it's something when you're asked to put up sometimes 10 to 20% of those numbers, especially if those grants number in the millions, right? You have to come up with cost share. So you have to actually be able to have money in the bank to provide against those grants. And I think most people don't realize just the extent and depth of reporting and bookkeeping that's needed just to manage, you know, not just one, but, you know, multiple is ridiculous. The learning curve that we undertook in those first 3 months was crazy because it wasn't just SBRs, it was a large DOE grant that's unlike anything I've ever seen. But yeah, so I mean, it's been great. It's really important to have all of that or have access to it, which is one thing I really applaud Oakcrest for providing as well. Is, you know, that infrastructure and the ability to get some of that, you know, grant management work, because it's not something that you necessarily think about as you go through that process. So it's more than just a budgeting exercise. So yeah, this is just some of us. You know, these were the first 4. We've now expanded beyond that. And we're always looking for good people to join our team. You know, if you know anybody within your network, I know Chris has sent some people our way, but we're looking for protein engineers, good technicians, people that have, you know, some experience with any kind of DNA technology, that kind of stuff. So yeah, we're not just looking for good people, we need to hire on some grants, so we have the money to do it as well. So yeah, thanks for your time. Thanks for having me. And I can answer any questions.
You do have one in the chat. Justine is curious if this the process that you're using is also how they make Beyond Meat.
It's not. So Beyond, I'm more familiar with Impossible's process. So like the Impossible Burger, that's basically a single protein that they express in, I think, yeast. It's the protein that gives it its red color. It binds heme. So to the extent that the technologies are similar in making the proteins, yes. You might not be aware, but all of you probably use enzymes every day. So enzymes are in your laundry detergent, your dishwasher detergent. They're what break up the fats and things like that. So enzymes generally can be really cheap. You know, we're talking about pennies per kilogram. The enzymes that we use are probably not so cheap, but still getting to those commodity prices, we can start to look at enzymes as they're coming into this realm of being true commodities, which is really good for us. you know, to empower a technology using, you know, commodities is great. So.
I think Brian has his hand up.
Yeah, I'm trying to figure out now. Do I lower my hand? Yes. No, I had a question. One of the things you said that caught me and reminded me of some interesting articles I've read in the past. Coming up with an engine to derive product, or coming up with a computational way to put these things together to derive product, begs the question in my mind of patentability and what parts become patentable. Are there risks as we go down these methodologies producing product that will just be seen as algorithms producing product and maybe not be patentable? Or how are you approaching those kind of problems?
I think so. It depends. So a lot of times there's not a perfect match to a biological reaction that does the given step that you're looking for. So in the case of the cannabinoid engineering, we had to take a natural protein that did a reaction similar to it and change it. So that enzyme now becomes patentable, right? Because it's not found in nature. If you're making natural compounds, those compounds often there's difficulties in patenting them. But to the extent that you can make non-natural compounds as well, those actual molecular entities should be patentable as well. You know, sometimes how you link those pathways together, if it's a non-natural way to do that, then those reaction schemes and those pathways can also be patented. It does beg the question of how defendable it is. And sure, that is a topic that we definitely understand and think about quite a bit. You know, there's always this debate about trade secret versus, you know, patenting. How likely is somebody else to come up with the method that we're using? And so, yeah, some of the core technology, because it's non-natural, you know, any of that non-natural type things should be patentable.
Okay, so
Hopefully that answered your question.
there's another question in the chat I think I saw.
There's a couple from Jeanette, and there's a number of them. Jeanette, why don't you go ahead? You were in there first.
Okay. Does this technique make it safer to work with potentially dangerous or infective cells?
Yeah, so we actually don't use any, like all of our things are BSL-1, which are completely safe cells.
Mm-hmm.
And so we don't have to actually deal with any cells. So
I mean,
the answer is
safer
that,
than traditional.
There's no pathogens, yeah, that kind of thing. I mean, the proteins that we use should be isolated enough away from the cell. So even if there was an issue with, you know, what are they, endotoxins from E. coli is sometimes an issue. It shouldn't be there as well. So this should lend itself. There are proteins that are used in GMP processes that have been derived in E. coli. So that shouldn't be an issue.
And another question I have is, in terms of techniques for innovation, how does this compare with, say, testing on digital twins? I mean, obviously digital twins don't have byproducts, so that's one major difference. But is it a lot faster than digital twins, or cheaper, or less so? Or
So I don't actually know what digital twins are.
So like a digital twin would be, you create the processes in a algorithmic mathematical model. And you're seeing this a lot with drug testing now for different compounds, that they're testing them on synthetic humans before they're testing them on animals and things like that.
So this process does lend itself to computational design. It should make it a lot easier and more streamlined. And so one of the things that it's kind of more of a premise. We haven't really tested this out. What most people do, they use these cell-free systems to prototype, meaning that they troubleshoot various different steps, get those steps working, and then pop them back into a cell, right? Which almost seems counterintuitive to us, because you've already worked everything out, and then you're going to put it back into that black box where there's a lot of other stuff going on. That's not what you've designed for. So it should be more streamlined in this. We have done a little bit of modeling, and our results tend to agree with those models quite well, which is really nice. And so the short of it is yes. I mean, if you've designed it, computationally and that pathway works, then I don't know why you would go back into a cell. Most of the time, there are some reasons why you might have to, but yeah, that's part of the beauty of this is it should be a much more streamlined, a much faster process as well. So, where it might take, like 100 person years to engineer a microbe to get it to do what you want, we should be able to do the same thing with like 2 or 3, right? And so that we have shown is that we can see better, as good or better results using a small number of people using this pathway and this technique compared to the metabolic engineering efforts. So.
Rolando has a question. He's apparently looking for 100,000 liters of cannabinoids. So if you unmute Rolando.
Isn't everybody?
Yeah, Tyler, thanks for sharing. I was just curious about the scale up and the yields for something like this. So are you if you're making cannabinoids, like the cannabinoid market, I'd imagine is what people want is kilograms of
Sure.
CBD or something. And are you going to be able to make that with this method?
Yeah. So the short answer is yes, absolutely. And it's really interesting that when you compare, there's 2 comparisons. The first is to the plant. I mean, the plant is so good at what it does. It's a weed. It grows everywhere. So for recreational use, it's really hard to compete with, if you're making CBD, THC, in the short term, it's going to be really difficult to compete with that. For a lot of these metabolic engineering companies, there's some unknown. So one of the things about our process is it doesn't scale like a fermentation. There's a lot of, you drive down the 210 and you see the fermentation facilities for, was it Bud Light? Miller Lite, I think. And you're like, oh, well, this is all easy. Like it's, millions of gallons. But a lot of times, like these processes fail when you scale up above, to the thousands of liters and they take time. We believe our systems are going to scale more like chemical reactions. So we do have data that suggests that it does, where we get the exact same parameters over 1,000-fold increases in volume, right? The other thing is that we can get to really high titers because just like a chemical reaction, if you just add more catalyst, things go faster and they can be more concentrated. So that also happens. And so we can now get the same amount of products in less volume, right? So your infrastructure needs are much lower as well, right? In addition to downstream processing concerns, where you no longer have to deal with the schmutz of a cell, right? Which is real gummy and messy and things like that. We have a pretty clean system. It's a couple enzymes with a little bit of buffer as a chemical and some other stuff. And so imagine separating that out, compared to the whole cellular milieu. Like it's much cleaner and it's really fast. And so, at the end of the day, we can get to more relevant titers faster at higher volume and more consistently as well. Not to mention that making different compounds is just as easy as putting in a different input as opposed to having to engineer a whole new strain. So yeah, there's a lot of room to grow in the cannabinoid space. Hopefully that answered your question.
Yeah, thanks. And by the way, I'm with you beyond cells.
Who did you work for at Stanford? If I can.
Yeah, I was in Drew Endy's lab.
Oh, you were? Okay.
Yeah, and so I I'm really familiar with this kinds of tech, and also a fan of other folks down here in up in Caltech and working on this kind of stuff. So it's very interesting, and just really excited for you.
Yeah, my email is right there. If you're interested in moving back here, reach out. We can talk further.
Will do. Thanks.
Yep.
Nice place to live.
Jim Schaefer wants some prognostications. Jim, are you unmuted? Yes.
Yeah, I'm not sure I understood like the potential products or applications in the near time and maybe in the far, the long-term future. What do you see as the products and applications for someone like me that doesn't quite understand all of the technical aspects?
So in the near term, the most likely targets are going to be therapeutic pharmaceutical compounds or intermediates that are really difficult to produce by other means, either chemically because they're synthetically difficult to get to, or because it's really hard to do that in cells. Cannabinoids are.
Can you give me an example when you say therapeutic? What do you mean?
So THCVA, which is the varin of THC. So the compound THC, which people smoke from, it's the active component of marijuana plants.
Okay.
So there's a separate analog called THCV, which just varies in one little area of the molecule. Okay. So it's if THC is 100% of what a plant produces, it does make some THCV, but it's at less than 1%, much, much less than 1%. Right? So isolating it is really difficult. Getting there from other routes is really difficult because that compound is difficult to synthesize, and there's just a variety of other reasons. So we can get to that compound actually quite easily. And so being able to do that, plus other derivatives that are like that compound, we can get to very quickly and very reliably, just by changing input. So it's still the same platform, but we can make a variety of different analogs of that very quickly and very easily at relatively the same titers. So the same amounts. And so it's very
long
powerful.
term.
And so long term, as we continue to develop and get better at, making the system more robust generally, some things need to happen hand in hand. We need to decrease the cost of some of our inputs, like the enzymes. But if you can do that, and it looks like we probably can, there's no reason why commodities are not on the table. These are things that are like plastic building blocks, things like that, things that come in at, anywhere from $10 to $50 a kilogram, right? Those types of price points. In the more long term, we're talking 5 to 10 years, there's no reason why biofuels are not also on the table. So, that part of it needs to be, some engineering challenges need to be worked out, but we're getting close to the titers that are needed to do that already. So, in terms of a reasoned approach, the higher margins make sense to go for first. There's a variety of other compounds as well. Astaxanthin, it's one of these compounds that they put in fish feed to turn salmon pink. It's actually one of the largest cost drivers of aquaculture for salmon farming. It's a really expensive molecule. It's also used as a nutraceutical, so you can buy astaxanthin supplements. There are some cells that make it naturally, some algae, but again, you have to separate it from everything. So we have routes to get to that compound as well, at comparable or better titers to anything else that's out there. And so yeah, going for these higher margin, lower volume compounds initially, and then broadening our, product portfolio, through a variety of, investment through government grants, in addition to partnerships with some of these bigger commodity manufacturers and users as well.
Thank you.
So
Are
It
you
was
going to be able to make?
You
Oh.
were bringing up the biofuels. The French fry car seems to have been displaced by electric vehicles.
Yeah.
What is the trend for biofuels these days?
Yeah. So I think, biofuels is really interesting because I think you're never going to see a scenario where liquid fuels are completely gone. And part of that is because you're probably not going to fly planes on batteries. You're probably not going to, sail ships completely electric as well. So there will always be some need for liquid fuels. And And so, to the extent that like the hydrogen economy and electric cars and batteries make all of our residential use, that I completely agree, and it should. But I think there is definitely a place for liquid fuels. And the good thing, a biofuel such as isobutanol, which is one of the projects that we have, it's not just used as a fuel, but it's also a commodity. So it's a building block chemical to get to a lot of other things that I think one of the things that people don't really realize is that the petroleum industry generally is not just about making fuels. Pretty much all the plastics that you use and that are in your things come from the petroleum industry. So to the extent that, we can make bio-based intermediates that can be turned into all these other products as well, it's a good place to be. So, there are methods to turn isobutanol into like isobutene, which is then an intermediate for polymer formation. So, these types of things and these types of projects are things that we're doing.
Brian Quant, you had a question about steroids?
Yeah, actually, I just watched a movie, and I'm for the life of me, I can't remember the name of it. I think it was The Life of Percy Levant Julian, and steroids came to mind as to, I would assume, completely applicable also for steroids. But I don't know, is it financially viable to do that?
Potentially, from a pathway and a feasibility perspective, steroids are difficult. There are some problems that we haven't found the best solutions to yet. But yes, we could probably build relatively easily the steroid scaffold. What's interesting is that, and difficult, is that for a lot of these natural products, steroids are one good example. A lot of flavors and fragrances are also the same way. It's not just the hydrocarbon. What happens is the microbe or the plant will make this hydrocarbon scaffold and then decorate it to make it more soluble, to make it stay around longer. And it's often adding those decorations, which is the really, really, really difficult thing. So, we have designs on doing that, but it's a problem that nobody's found a good solution to yet.
Go ahead, Paul, if you had a question to follow up on the steroids.
A question, just a comment. I thought steroids were fairly inexpensive products to make.
Well, there's that too. Yeah. I mean, it's, expensive is also relative, right? We try not to, and that was one of the problems that we had early on is this idea of getting into a race to the bottom. For flavors and fragrances, that's almost the game. And one of the problems there, if it's easy to get to, say, chemically, people don't pay much of a premium for bio-based, for instance, right? In flavors and fragrances, you also have this problem of biosimilars. I don't know if it's so similar for steroids as well. But if it's just good enough, probably not so much for the steroids. But for flavors and fragrances, if it's close enough, usually, and it's cheaper, then they'll go with it, But yeah, you're not wrong. I mean, it all comes down to what's the price, and those prices are relative. I mean, if you're saying steroids are $1,000 a kilogram, is that cheap? I mean, maybe yes, maybe no. Depends on your perspective.
Bruce, go ahead.
Yeah, so on the SBIR grants, I have a couple of clients that have SBIR grants, and how helpful is the government or forgiving when you're trying to get into this? Is it sink or swim, or did you get a lot of help, or at least get the sense you had help in getting started with these grants?
So the SBIRs they weren't necessary for us to get off the ground, if that's the question.
Well, no, I guess the question is, could you turn to the same people that could cause you grief? Were they helpful to help you stay out of trouble in terms of dotting the I's and crossing the T's and doing your accounting, all the things that you were saying were so difficult, did you get help from the government or were you on your own?
We were on our own. I will say though that going through that exercise has made the company generally better. You know, a lot of times, you know, business accounting, I don't know much about it, but the government is so rigorous in all that they do that, you know, we are now top to bottom with everything, you know, timesheets and everything that is accounted for. I mean, it's, you know, every dime is accounted for. So
Okay.
every penny. I mean, yeah, it has made everything better, but you know, the government basically contracts with like Jameson or something like that, right? Which are really, they're these contractors that will help you manage grants as well. They're one example. I think they tend to be really expensive. You know, we had potentially a couple other individuals that could have helped us as well, but they were just through word of mouth, right? We knew other people that had SBIRs, or who do you use, that type of thing. But no, it was not the government that put us into contact. I don't know if they would have if we had asked. It wasn't a question that we did ask. You know, one of the things that happened to us was that the SBIRs would have been manageable, and we would have been able, like with our finance team, we would have been able to figure it out. We got this large DOE grant, which was a 3-year, $3 million grant that had cost shares associated with it. It had all of this documentation, not just on the accounting side, but on the PI side as well. It really is a massive amount of work, and we really needed help. And so we were lucky that Oakcrest had kind of, I think the idea is that it's going to be part of the offering for the incubator is to offer some of that support. I don't know how many incubators offer that. It wasn't necessarily something that I believe PBC, I think I saw Wendy on here. They offer some help with that, I think, or at least individuals that can help with it. But yeah, the person that we have now, we were put into contact with through Oakcrest, and she's been great.
Good. So I had another question on the biofuels and the plastics. Can biofuels be engineered to burn cleaner? Can plastics be engineered to biodegrade better with some of the things you're doing?
Yeah, so the bioplastics probably. I mean, I don't know about, you know, the burning cleaner. That's downstream from us. That's more on engine design. But you know, bioplastics, I think, you know, they are bio-based and they're made by enzymes. They can be broken down by enzymes. I think that's generally the idea. So the short of that answer is yes. You know, I did also just read an article, I think it was a LinkedIn post maybe, that researchers at NREL, so one of the national labs, have found microbes that actually eat plastic, like they degrade polystyrene. You know, so nature has found a way. So sure, to the extent that you can build it up, you can probably break it down biologically as well.
And if there's no one else, I have another question. I know when I first talked to Chris about the incubator, they were involved with a VC or something like it. Is that the same VC that you're using, or did you have your own lined up as you came in?
No, we had already lined up our own private funding prior. We ended up going with one. We had another that was on the table as well. And they did not come together, the two sources of investment. And so just prior to us signing the deal, it was looking like it was likely enough that we were going to need to find lab space. And so we started looking. And so we already had our money lined up and we knew that it was going to be there. We started looking for incubator space. And, you know, incubator space is it's a really interesting thing if you think about it, because if you have a small SBIR or maybe a small seed, right, 250,000, maybe 500,000, in the biotech space, it's really hard because centrifuges, -80 freezers, these are expensive pieces of equipment. that you can't necessarily pay for with a $250,000 budget if you hope to pay yourself as well, right? And for us, we knew we had other things that we wanted to do, and we, a 6-foot lab bench wasn't going to be enough. And so there's actually not a lot of incubator space out there that is in this in between for 2 to 5 people, this ability to find 600 square feet to house these people, a little bit of equipment, some shared equipment. We were lucky that we came with our own money so we could outfit the lab. But I know that not all biotechs are necessarily going to be like that. And so, I think the biotech generally is a tough nut to crack, because there's, specialized equipment and that was one of the problems with some of the other incubators that we looked at, is we knew it wasn't going to be enough space in the short term. And we knew we couldn't fill up 3,000 square feet, right? So where's this intermediate amount of space going to come from? And so Oakcrest was actually just perfect. It was enough space for us to grow, to do what we needed at the time. So, I think there's something to be said for a place that's set up by scientists. Like they know what scientists need to do science, especially on the bio side, instead of just putting a shell with a couple benches and saying, "Hey, figure it out." So that was really refreshing. So I think those things tend to exist a little bit more in the Bay Area. I know there's some in San Diego. It's just LA is tough. They're few and far between.
So Tyler, in that discussion, what are we missing? We started that conversation, just the two of us, a week ago. But what do we need here to anchor the cluster? What's missing? What are the pieces?
Yeah, I think buy-in from the San Gabriel Valley biotech generally, like LA biotech, this ability to network and to bring, multiple people together. I mean, it's hard right now in COVID, right? Because there's not those abilities to network, but I know that's when we've talked about this, that's one of the big things up in the Bay Area is, this ability of people from a variety of different companies to come together that do similar enough things that, they can talk shop, they can, network amongst themselves. I mean, that it's hard to do in LA because it's so spread out, but, if there is this, we don't see many people from City of Hope. We don't see many people out in Monrovia. I don't even know of many events besides MADIA, that are kind of geared towards that. So I don't know, as things open up more, when I was at UCLA, we didn't see, I only became aware of MADIA and some of the stuff in Pasadena because I lived around here. But at UCLA, they didn't advertise for it. But you might be surprised at how many PhD students, postdocs are interested in the business side or interested in startups that are at the universities. UCLA has a business and science program. To link into those, I would be surprised if USC didn't as well. To reach out and let these universities know that there are these opportunities around to network. I always said at UC Irvine, I always wished that there was more support. It's this type of thing. You're in your lab, you're going to graduate. And if your boss is not well connected or whatever, it's like, good luck. So, to the extent that you can kind of mine some of these resources, I think, there's people out there that
it's very hard to get that first step going, right? You have to attract enough people initially to attract more.
Yeah, and it might just be also like tapping the right people that are at these universities that are, invested in seeing the people there move on to bigger and better things that are not necessarily just professor gigs, right? I remember early on, one thing that started at City of Hope this was when I started UCLA in 2008, but then I think it fell by the wayside, was there was a professor there who was a crystallographer who was trying to bring together Caltech, UCLA, City of Hope, all of these people doing biotech type stuff. I looked him up the other day. He's also started 2 of his own companies recently. So, you know, he has that, somebody like that, you tap him and be like, look, there's this network. Like, how do we get those types of individuals that are involved? People at USC, I know that there's a couple people in like the chemistry and the pharmaceutical sciences that are these serial entrepreneurs that, you know, might be interested. It might just take enough of tapping the right couple people to get it started. If even just to disseminate, you know, to other people. I don't know. It's just an idea. I don't have the perfect solution. I try and tell everybody that I come in contact with about how great this is. But that's not a lot, and my kids don't care.
I think that maybe the power of networking is lost on young people until they're finally get old enough to realize it's all about your network.
Yeah.
Especially with scientists and engineers, they're not really natural networkers. So how do you get them to come to an
Well,
Yeah, I got a question. Is it, Tyler, is it probably, does the idea stem from that the university is trying to keep the ideas to themselves so that they, you know, people like you can stay within that structure, and they don't have to share that with because as you stated earlier, when you left UCLA and it cost you to get that idea back for yourselves. So if you wanted to participate, you had to pay the university for that technology. So,
well, if we wouldn't have let the university know that somebody else was interested, it would have been great for us because they would have just let us. The universities do want their inventors and the people developing the technologies to spin things out, and they are generally supportive of that. You know, I mean, at the end of the day, they do own the technology. You have to license it back, but I don't see that being the biggest problem for most people starting. I mean, it's, you know, a lot in the biotech space, it's having that good hook and being able to raise outside investment. Unless you come from a situation where you can raise some friends and family round, you know, type thing, or you get lucky with an SBIR. But yeah, I don't think that's necessarily so big of a problem. And I will say that I think that so I know UC Berkeley does a fantastic job about it. UCSD I think does a really, really good job. I don't know, UCLA is trying. And I think it's, you know, within the past 5 to 10 years that some of these universities have, you know, come around and really embraced this kind of startup environment. I think the LA schools have just been a little bit behind. You know, I think you can tell part of where the hubs have started is, you know, you can see that the universities around there have really embraced it. Stanford, they have great deals for their professors. You know, they can be CEOs of companies at the same time they're PIs. That doesn't happen at UCLA or any of the UCs. So, you know.
So one technical question that I wrote down earlier is what you're doing going to replace cell-based synthesis, like biologics? Is it?
So probably not for biologics. Although that being said, there is a company up in the Bay Area that was based off of technology developed at Stanford that does make antibodies cell-free. It's called Sutra. So Jim Schwartz at Stanford, he developed that. So it is possible. That's not what we're trying to do, at least not for biologics. For other types of commodities and some of, you know, petroleum-based things and difficult to make natural products, the answer is probably yes. That's what we're trying to do. But, you know, that being said, cells are really good at making some things. There's some things that you probably would never really want to do cell-free. You probably wouldn't want to make ethanol because yeast have been, you know, evolved over thousands of years to do that, and they do it really, really well, right? So, you know, picking your place is also really important. Like, you know, you're not gonna really compete with cells to make amino acids or some organic acids, right? So
Okay, and got any more questions, and we can, we're on the hour now, so any last questions from anybody?
I see none on this chat.
Okay, well, thank you very much, Tyler. That was very informative and exciting that you're doing it in Monrovia and at Oakcrest. Hopefully it'll be Oakcrest's first home run outside of what they do normally.